Hello, my name is Brio Hamal and I'm a student in Genetics 311. Today, I'll be going over my Exam 5 mini video. I chose prompt 1, so for my Exam 5 mini video, I'll be going over the adapter hypothesis. So this is the illustration I came up with, the diagram I came up with to show the adapter hypothesis. I think the name adapter hypothesis relates to the known function of tRNA because the tRNA molecule attaches to the mRNA ribosomal complex to add the appropriate amino acids to the growing polypeptide chain. So by bringing the appropriate amino acids to the ribosomes, the tRNA acts as an intermediate in building the appropriate polypeptide. So tRNA molecules, you can see here, have anticodons. mRNA molecules have codons. These tRNA anticodons are complementary to the mRNA codons, allowing them to bind. So, for example, you can see here the GGGs are able to bind to the CCCs because they are complementary. And tRNA is bringing all these amino acids to the ribosomes to make an appropriate polypeptide. This is the process that occurs to create appropriate polypeptides. But alterations can occur. An alteration that can occur is a mutation in the codons. So a mutation in the codons can result in tRNA not being able to bind to the codons and the mRNA. With no binding being able to occur, the result will not allow protein synthesis to occur properly. So protein synthesis not being able to occur properly can lead to many cellular functions and metabolism of cells being affected. When cellular functions and metabolisms of cells are affected, cells may not be able to survive. So we can see that tRNA is very, very important in the process. This was my diagram of the adapter hypothesis, and I appreciate the time. Thank you and have a great day.